I think it's time to start.
Yeah.
It's my pleasure to introduce Professor Leon Glass. Dr. Glass is a professor and Isidore Rosenfeld Chair in Cardiology at McGill University. He was born in Brooklyn, New York, and obtained a PhD in chemistry in 1968 from University of Chicago by studying the theory of atomic motions in simple liquids. In 1975, Dr. Glass joined the Department of Physiology at McGill University. He was awarded the Guggenheim Fellowship in 1994 and is a Fellow of Royal Society of Canada. Also the American Physical Society and the Society for Industrial and Applied Mathematics. Dr. Glass carried out research on the application of nonlinear dynamics to physiology and medicine, and many best be known for work suggesting that certain physiological disorders may be considered dynamical disease. One example is understanding the mechanism that leads to transition from normal heart rhythms to pathological rhythms. Something I'm also closely interested in lately, the beating in unison. Today his talk is entitled Evolution and Robustness in Genetic Networks. May I introduce Dr. Glass? Thank you very much.
Okay. So thanks very much, and I really had a great time here yesterday and today, and really enjoyed meeting a whole bunch of interesting people. So thanks so much for the invitation. So I start with this slide, and this is from a paper published in Nature about 5 years ago, and it says transcription factors bind in a combinatorial fashion to specify the on and off states of genes. The ensemble of these binding events forms a regulatory network constituting the wiring diagram for a cell. Okay, so that's the basic point. So do people here, I'm not sure whether, I know there's a mix of mathematicians, a mix of biologists. People can feel free to ask questions if things are unclear. Do people know what transcription factors are from the mathematicians? Okay, so what they're saying is that there's a combinatorial control of gene expression, on and off, suggesting Boolean logic. So this is a very, very specific question, and I'm very, very specific statement, and I'm not sure if this is correct or not correct. I'm not a co-author of that paper, but maybe one of the people in the room is a co-author of the paper. Is Alan? So you're a co-author of that paper. Okay, so is that statement correct, or? Okay. He stands by the paper. Okay, so, you know, they have these very complicated diagrams, which is the wiring diagram, which they figured out as the control network, which transcription factor is binding to which thing. Whenever I see a picture like that, I always raise certain doubts in my mind about what the lines mean, whether I should believe the particular line, and so on and so forth. So I would ask the question, is there a simple way to think about genetic networks? If no, then there's a big problem because there are 20,000 different genes, more or less. If yes, then is it possible that ideas based on what they were saying, that combinatorial on and off networks, may play a role and may be relevant? Okay? Now, going back before that, we have this other quote, which is from Indika's friend and colleague, Hardwell and colleagues. The next generation of students should learn how to look for amplifiers and logic circuits, as well as to describe and look for molecules and genes. So they're saying essentially the same thing, that let's look for logic, let's look for logic and control of how genetic networks work. Where did these ideas come from? Okay, so I think they came from here. So this is a classic article by Monod and Jacot, who were the original people who discovered transcriptional control. And they say these examples should suffice to show that by the use of the principles which they illustrate, any number of systems may be interconnected into regular circuits endowed with virtually any desired property. The essential point about the imaginary circuits which we examined is that their elements are not imaginary. Okay? So they're showing, in this case, they're showing how you can put together regulatory genes acting to how you can put together control genes. This is a picture of what they would say is a mutually inhibitory network that could be used to build a switch. So they were suggesting that these networks could underlie such processes as differentiation, oscillation, and so forth. Now, in the talk, I'll give a short history of logical models of gene networks and summary of the main results. I'll talk about robust dynamics in gene networks and also evolution in model networks. Now, a lot of these ideas, I think, go back to early papers by McCulloch and Pitts talking about neural networks. Then, in terms of neural networks, there are a series of papers following that by Kling and Sekeli, Jack Cowan, Terry Sosnowski, Steve Grossberg, Hopfield, and many, many other people talking about logical models and developing the whole field of neural networks, which, as people know now, is very much a major area of research. Slightly later than that, there was talking about genetic networks. There was the paper by Monod and Jacot that I just talked about. And shortly after that, there were papers by Sukita, Stu Kaufman, René Thomas, Gray, and many, many other people subsequently. I learned about these ideas initially from conversations with Stu Kaufman. Stu Kaufman was at the University of Chicago as a young faculty member when I went back there to do a postdoctoral fellowship. And Stu had this graph in his paper in Journal of Theoretical Biology back in 1969. So this was something that I thought was completely fascinating. He was making up networks of Boolean switches. He was having sequential updating, random functions of Boolean networks with two inputs, randomly chosen from the network. And then what he showed is that the number of attractors that you have in those is comparatively small compared to the number of states. If you have 100 elements, you have 2 to the 100th states, you have an astronomical number of states, but the number of attractors was comparatively small, of the order of 10, and the length of the cycles of those attractors was also comparatively small. So Stu's notion was that we had these attractors. The attractors could be associated with different cell types, and instead of wandering around randomly in phase space, the way you tend to do in statistical mechanical systems in physics, you were actually focused into a comparatively small number of attractors. So there are many details in this particular picture, and I think that my opinion is actually most of the details are not correct. I was talking to Stu Kaufman over the summer, and he says, well, actually, most of the details really are correct. We won't debate that at the moment. But he was basically saying that the number of cell types is in some sense related to the number of genes. Okay? So to me, that was a fascinating notion. Now, we collaborated in writing a paper called The Logical Analysis of Continuous Nonlinear Biochemical Control Networks, in which we made up equations and had a model, which is going to be the main point of this talk, to try to present that. One of the things which is fascinating to me is that this paper was published in 1973 and for the next 20 years was basically never cited. Okay? And then it became much more popular subsequently. So anybody You know, like I was talking to some of the people about career evaluation today and graduate students and so forth, so forth. So if you're a chairman of a department and you have somebody coming up and they say, well, I wrote this great paper, but unfortunately nobody ever cites it, you can say, okay, try to read the paper and decide if it's really a good paper or not, instead of just counting the citation. Anyway, so it doesn't mean that it's cited, it's a good paper, but it means that at least people cited it. Okay, so I'm just going to go through a few slides. So the notion of having logical networks has been used by many, many people. So one cannot, I can't give all of the people who have done this style of work, but I'll give a few of the earlier papers. Denis Tifri and René Thomas were proposing the same type of mutually inhibitory network that could control a switch in bacteriophage lambda. Eric Davidson and his group, over many years, and particularly with Hamid Balouri, were developing largely logical models to control differentiation, which was based on extensive experimentation, particularly in sea urchin. And then Elena Alvarez Villa was studying logical networks that were controlling gene regulatory networks in Arabidopsis. So there are many, many models of this style that have been proposed along the way. The details of the particular models are often different. Many of these models have this updating in discrete times, where you have discrete switching of the logical network. Okay. Move forward to 2000, and there were two papers that came out back to back in Nature in the year 2000, which many people here are probably familiar with. One of them was by Jim Collins and colleagues, in which they actually built a circuit which was based on the mutual inhibition, where there's the two represses. One transcription factor represses the production of another transcription factor and vice versa, and depending on which one wins out, which one gets in front of it, it will suppress the other one. So this is a primitive model of differentiation. Once again, exactly the same idea that was in Monod and Charcot, but they get the credit in this paper and the paper that follows for actually having started the field of synthetic biology by actually building it. So they did experiments and they demonstrated that it basically worked. Here's a slide that I show and that probably nobody here has ever seen. This is from a 1968 paper by two Hungarians on rhythmic neural activity, in which they said, here's a little circuit of three elements, each one of which inhibits the other one. This is a model for oscillatory neural systems, and here's the activity of three nerves that they made. So maybe they even made some analog systems that had these sorts of behaviors. So they developed this network of three elements, mutual inhibitory network, and then in that same issue of Nature, Michael Elowitz and Stan Libra designed an oscillator of three mutually inhibitory factors in which there was a generation of oscillatory behavior. And then they demonstrated that. And this was the other co-paper that's really the founding papers of this area of synthetic biology. Now, this is the result of a generation of the same circuit that we generated. This was generated during the year that I spent as a sabbatical with Jim Collins in an electronic circuit, and I'll tell you more about the circuit towards the end of the talk. But the basic idea is that three elements, one inhibits two, two inhibits three, and three inhibits one. And one of the ways we think about this is in terms of an underlying logical network. And in the logical network, we would say if one is inhibiting two, then if one is high, then two is going to be the logical function will be 0, and if 1 is low, 0, then 2 is going to be high. So that's the logical function that we associate with inhibition. And when one goes and does this for all 3 elements, 2 inhibiting 3, so 3 is an inverse of 2, and 1 is an inverse of 3. So this, in some sense, represents a truth table for this logical structure. So that's how we go from the logical structure to a truth table, a picture like this to a truth table. Now, the basic idea, or one of the basic ideas behind what we've done, is to now go from the truth table to some transition diagram. Transition diagram would basically allow transitions in some classes of circuits that represented this logical structure. What do I mean? If we're in the state 0, 0, 1, in other words, 1, 2 are low and 3 is high, then in the next time step we would go to a state 0, 1, 1, where 1 is low and 2 and 3 are high. So this is just following from that truth table. This is just a representation of the truth table. In this case, we would have an edge directed from a state 0, 0, 1 to 0, 1, 1. In similar fashion, we would go from 0, 1, 1 to 0, 1, 0, etc. However, if we start off at state 0, 0, 0, think of what's happening. 1 inhibits 2, 2 inhibits 3, 3 inhibits 1. Nothing is inhibiting anything else, so all 3 can go high. But in any realistic version of it, only one thing would go high at the next time. You wouldn't have the situation where all 3 went high at the same time. So in this diagram, in the state transition diagram, you go from 0, 0, 0 to each of the 3 possible states that could follow next. So we generate a state transition table based on a state transition diagram as a directed graph, in this case on what would be called the 3 cube, from the truth table. Okay? Okay. So what's the idea? The basic idea now is to take the logical network and embed the logical network in a differential equation. And in the differential equation, we have continuous variables, and associated with each continuous variable is a logical variable. What does this mean? So here we have a continuous variable, x1, or is it xi? x1 may be going up and down. If x1 is beneath a threshold, then the logical variable associated with that is going to be 0. If x1 is above a threshold, then the logical variable associated with it is going to be 1. When x1 goes from 0 to 1, if x1 is now an input into some other element or some element of the system, then the function, the logical function which controls what this other variable is doing, can switch from being high to being low. And this is basically the differential equation. So the differential equation assumes for each variable there is an exponential decay. And then there is also a control. And the control is represented by a logical function where the continuous variables are translated into Boolean variables. And then this logical function is going to go on and off depending on exactly what the logic is of the thing. So is that equation clear? So I'd like people to understand what that equation is, even if nothing else is clear. Okay? So, and this was an equation that was worked on and developed by myself, Stu Kaufman, and Joel Pasternak back in the 1970s. So the rationale for the equation is very important. So in biological system, we often have diagrams in which we say one thing activates or inhibits another, some kind of wiring diagram of activation and inactivation. And we need a way to represent those diagrams. So that's one type of knowledge that we have, can be measured generally. You can also measure patterns of activation and inhibition. Often those measurements are done with a huge amount of noise and not very reliably, but in many cases you can get patterns of activation and relative patterns of activation that's present. So these equations enable us to relate the relative patterns of activation with the underlying structure of the network in a very crude way. The other thing is, which fascinated me, is that the equations also allow detailed mathematical analysis, which involve discrete math problems, also issues of nonlinear dynamics, dealing with stable limit cycles, chaos in higher dimensions. So because of their piecewise linear nature, these equations are amenable to actual mathematical and theoretical analysis. Okay. The representation I showed of the repressolator, the network that was built by Elowitz and Libra on 3 dimensions, but this is extendable immediately to any network in n dimensions. So the state transition diagram for these systems can be mapped onto an n-dimensional hypercube. An n-dimensional hypercube has n vertices, has 2 to the n vertices. It has n times 2 to the n minus 1 edges. And each of those edges represents a transition from one logical state to another logical state in some associated differential equation or other updating scheme, like a stochastic updating scheme, where one thing would change at a time. For networks with no self-input, there's a corresponding directed n-cube in which each edge is oriented in a unique orientation. So if we have an n-dimensional network where nothing turns itself directly on or off only through an intermediate, then any given network can be represented by a directed graph on an n-dimensional hypercube, and going backwards, we can go from the n-dimensional hypercube back to the logical structure of the network. So it can go both forwards and backwards, and then that representation is valid in a variety of different circumstances. Okay, so in terms of the dynamics, talking about robust dynamics. Okay, in this particular example, there's a cycle on the 3 cube, and this cycle is attracting, meaning each edge that touches the cycle that's not on the cycle is adjacent to the cycle. So that's a structure which we call a cyclic attractor. And there was a theorem that was done with Joel Pasternak that says any attracting cycle on the n cube is associated with a stable limit cycle oscillation or approach to a stable fixed point in the associated differential equation. And we also, in most cases, as you go up to higher dimensions, it's easy. The stable limit cycles really prevail. So this is a result which asserts stability and also uniqueness of limit cycle attractors in n-dimensional dynamical systems. So from the point of view of nonlinear dynamics, once you go up to dimension higher than 2, it's very hard to prove stability and uniqueness of limit cycle oscillations. And from a mathematical point of view, we did that in this particular equation. And aside, if you take the step functions and turn them into sigmoidal functions, you also tend to preserve the stability and uniqueness, but that's not as well proven, although there are proofs moving in that particular direction. Okay? With a graduate student, Roy Wiles, we developed a concept which we called minimal networks for robust dynamics. So the thing about these attracting cycles is that they're associated with stable limit cycles, and stable limit cycles are robust. They tend to be stable. So if you have a network in which you have one of these attracting cycles in higher dimensions, then in the differential equation that you might generate from it, you would have a stable limit cycle oscillation. Okay, so one can come up with a procedure for building these networks. So this is associated in particular cases with building a network which gives a particular dynamics. So we can have a particular path through this discrete phase space on a hypercube. We can say all the edges are attracted to that particular path. Then we can fill in the truth table associated with that network, and then we can find the simplest functions associated with that particular truth table. As we go up to higher dimensions, the truth table will have many elements which have not been determined, but we can say if that cycle is attracting, then we know the values in many of the elements, and then we can make up simple functions for other elements as simple as we possibly can. So this is a way for going from an observed dynamic to say what network can generate that particular observed dynamic. There's an example that we give. So I know you can't see those equations, but when we go to a 5-dimensional system, if we have a 5-dimensional system, we can classify patterns of oscillation by the sequence of variables that change sequentially. So 1, 2, 3, 4, 5, 1, 3, 5, 2, 4 would give a sequence in which each one of those hypothetical genes would switch. And then we can say, can we generate a differential equation which would be associated with that particular sequence? And the answer is yes. And that differential equation will have a stable limit cycle oscillation. And in the differential equation, we can also, instead of having these piecewise linear functions, we can have sigmoidal functions, and we can study such properties as the bifurcation, change of stability, and the amplitude and frequency of the oscillation as you change the steepness of the underlying function. Okay. Ooh, wow, that's bright. So we have, so this is from a paper that was published in PNAS. The yeast cell cycle network is robustly designed, and what they did is they took 12 genes and factors that they thought were essential for the dynamics in yeast, and they had the same basic idea. They say that they have a pathway in this discrete space. Each of these dots represents a discrete state of on or off of this network. They say there's an attracting pathway which is corresponding to the yeast cell cycle that you have here. From talking to John Tyson gave us the sequence of states that are associated with that cycle. And then what we did is we went backwards from that and said, if we have the sequence of states associated with the cycle, can we come up with an underlying differential equation and logical functions that would generate this sequence of states without knowing anything about the biology? And the answer is yes, you can do that. And when you do that, you come up with a series of logical functions which turn out to be very close with what people would have called at that time the correct functions concerning what the control for each of these different transcriptional factors would be. Okay? Oh, wait, that's, so this one, oh, yeah. And then this represents what the trajectory is in that particular differential equation. So this represents what the dynamics would be in that differential equation. So this was just really a proof of concept that it can go backwards from some sequence. So in things that Indica does, if he has a sequence, if you can actually say, here's a sequence of things as they go high and low, what this is saying is you can go backwards if you say that that's stable, robust, you can go backwards and say, here's a potential interaction network that can potentially generate that. So that's a way to do the inverse problem. Okay? So then with Chava Siegelman, we tried to write down and formalize that in a current opinion article talking about how you can go back and do this inverse problem. Okay, in terms of evolution of gene networks. Okay, so in these networks, we're saying each one of these model genes gets inputs from the other model genes. So then we can ask the question, as we go up in dimensions, how many different networks are there? Okay, so remember that each entry in the truth table gives you, it corresponds with one of the edges. So, for example, on the 5 cube, there are 80 edges. N times 2 to the n minus 1, I think, would come out to be 80. 5 times 16. Okay, so you're in a system where there are potentially this astronomical number of ways that you can build these particular networks. So that becomes very, very big by the time you get to 5 dimensions. So this is the total possible number of networks it can have. But then you can also ask the question, under the symmetries of the n-dimensional hypercube, how many of these are unique? How many of them are different? So, for example, in 2 dimensions, there are only 4 different ways you can label the directed edges of a little square. So if you put in directed edges on a square, you can convince yourself very easily that there are only 4 different configurations. You can have a cycle. Anyway, you can do that quickly. You can place a lower bound on the number of different networks under the hypercube by dividing the total number of networks by the number of elements in the symmetry group of that particular hypercube. So when you get to 5 dimensions, the total number of networks that you can have, this is without taking into account the symmetry, is of the order of 3.14 times 10 to the 20th, and the lower bound is equal to that up until about 7 decimal places, or 6 or 7 decimal places. So the lower bound is very close to the real value. Mind you, there are trillions or tens of trillions or trillions of things which you're not counting, but these numbers are astronomically big. Okay, so that's just one of the things which is interesting that we can do in these because of their discrete structure. Okay, so we try to evolve long rare dynamics, and I'll say a little bit about evolving long cycles. So I'm supposed to finish by what time? 4:30. Okay, so it's still okay at the moment. We tried evolving long cycles, and we also tried to evolve chaotic dynamics. So in terms of evolving long cycles, so the backstory of this is that I was friends with Jim Collins. Some of you here know Jim Collins, one of the founders of synthetic biology. He had worked on things in cardiac arrhythmias and dynamics of walking and stability and so forth, balance. So we were friends from his times in those days. And after he did his paper on building the toggle switch, I went down to spend really a sabbatical with him, part of the time sabbatical, saying, well, let's try to evolve genetic networks that have a particular dynamic. So we quickly realized, or I quickly realized, that it was impossible in that period of time at that time to actually evolve genetic networks that had particular dynamics. But we were able to say, well, okay, we can't really we can't do it in a bacterium, but let's try to do it in an analog system. So a student by the name of Jonathan Mason designed with Paul Lindsay, who's a physicist engineer who was at MIT, designed a combinatorial switching circuit so that we could, for each model gene, we could read in a truth table, and then from that truth table, we would be able to generate essentially an RC circuit, so that if you were going high, you would be exponentially approaching the high value, which is 5 volts, and if you were low, you're exponentially going down to the low value. So it would be possible to feed into these circuits a truth table, which was 80 ones and zeros, representing the interaction matrix, and then to try to evolve dynamics that would be rare. So this is what the gene network looks like in that context, and this is what a gene looks like. Okay, so these are the old-fashioned kind of circuits, and then just doing a random search of random networks, trying to see what the periodicity would be just from randomly built circuits. Here, the RC circuit was built so that the relaxation time was of the order of 10 milliseconds, and there were very few networks that spontaneously had periods longer than 60 milliseconds. So the object was to try to evolve things that had longer periods than that. And then this is just an example of 3 runs in which, here are 3 different runs up here, trying to evolve things with periods longer than 75 milliseconds. And here's typical traces that happened in one of those runs as you are evolving this. So you have the truth table, you start off with something with a period, you randomly perturb certain fraction of those elements, and then you see what the cycle is. If it's equal to or longer, you're going to keep the network, and then you update it until you get a long network. So, that to me is kind of a fun project. Another project that was done, this was done now in silico, where Roy Wiles, along with Stu Kaufman, now took these networks with, just in a computer, and then did the same process of evolving the truth table in a computer. And the idea was to try to evolve something that was more irregular, and the irregularity is based, the measure of irregularity is based on something that's called topological entropy. And for people who don't know what that is, don't worry about it. It's just associated with how irregular the particular rhythm is. And then it was also possible to study the most effective level of perturbations. In other words, they're going to change a lot of these ones or zeros at any one time, or a few ones or zeros at any one time. And the bottom line of that is that if you're very far away from where you want to go, you would like to change more things randomly. And if you're very close to where you want to go, you want to change fewer things randomly. So that's just results that were on there. Okay, so all of this has dealt with things that don't have anything to do with biology in a very direct way. So I just like to try to mention a few things related to practical problems and practical issues. So there was this very interesting paper from Odell and colleagues talking about robustness and evolution in natural systems insect development. So here they say the segment polarity network is a robust developmental module. So I'll just show you a little bit of work that was done in this particular paper. And this is really, the analytical work was done by Ted Perkins, who is a postdoctoral fellow with us. And we collaborated with Jaeger and John Reinitz, who have developed neural network models for development of Drosophila segmentation. So for people who don't know about this, is that different genes are expressed at different times during the early developmental stages in Drosophila, and they have these beautiful banded patterns. And John Reinitz and his colleagues had been developing neural network models to try to reproduce these developmental changes. When I saw this work that John had done, first of all, I thought it was absolutely fantastic. It's beautiful work. But then the question is, could you do this inverse problem? When they did this, they have their neural network models. The neural networks are actually, in many ways, very similar to the genetic networks. And I can develop that idea further if people are interested. But they were doing the simulated annealing. They had very, very lengthy procedures to set what the different functions are in the neural networks and thresholds and weights and so on and so forth. And the notion that I had is that one could represent the same thing by logical networks, and that it may be much easier and faster in order to get the pattern. So Ted Perkins, for one thing, is that the basic class of models that it deals with are called reaction diffusion models, in which there's diffusion of these transcription factors, and also the production of the transcription factors is based on some particular function. And Ted developed a way in which to converge to the final weights and the final patterns much, much faster than was being done by John Reinitz, by in some sense breaking it up into a number of individual steps and stages. So this was done. This is what the basic data. So this is what the experimental data. This is what the different data is looking like in different classes of models, which have been updated and which have been converged to the final values. So many of these particular models, the neural network models or the logic models, just differ from each other in how well they fit the actual experimental data in terms of the number of stripes, the position, and the timing of them, only in very small ways. So many of these, one of the thoughts that I had is one could come up and the inverse model would be very clearly defined, but that is not what happened. There were many possible inverse models that were consistent with the data, and you would have to use other biological experiments in order to get that. One can come up at the end with an actual network, and the network in these cases has a very well-defined differential equation structure that's associated with it that can be used to do the simulation. And then finally, I would say there's another interesting question. So we also collaborated with Ahab Abuheath in Montreal, who studies development in ants. And he's interested in the evolution of the genetic networks controlling development in ants. And with a graduate student, Marcos Namad Ben Susan, we developed differential equations for these networks. And one of the main points that came from that is one can observe the phenotypes of the development. If you look at different ants, to me, they basically all look the same. The notion, you might say, well, what's different between the different species? And one of the notions is that even though the phenotypes are very similar, the underlying patterns of activation of the genes can actually be different in the different organisms. And this is something that they have called developmental system drift. Okay, so in the talk, I've given a little bit of a history of logical models of genetic networks. I've talked about robust dynamics in these networks. I've talked a little bit about evolution in model networks. There are many future challenges. A mathematical challenge, so a basic mathematical question that's really motivated me, in some sense more than the biologist, may be evident in the talk, is given a logical structure of a network. And each one of these logical structures is consistent with a whole range of different parameters in the differential equation. I just showed the simplest set of parameters. But each one of these is really a whole equivalence class of different models. You can change production rates, decay rates, thresholds, etc., all with one of these structures. So given a logical structure, what can we say about, how can we read from the state transition diagram all of the possible dynamics that you can have with a given state transition diagram? So that's one of the challenges in the mathematics. That's one of the challenges in mathematics. From the biological point, I think that the question of how do we understand the structure and the evolution of genetic networks in organisms is an amazing question. I was telling Indika yesterday that most of the work that we do is in cardiac electrophysiology. If we look at the hearts of mice and men and elephants, they all have a very similar anatomy. They all have very similar ion channels. The frequency of the beating is very different in the different organisms. But there's something very robustly conserved in the evolution of the network. And I don't know how that can possibly happen. Okay? So I think that's an amazing question of how real genetic networks are really evolving. And then I think that this question that we raised with Jim Collins, how can we actually evolve real networks, but in model organisms to do particular things, that's something I think that people are probably working on that now. Okay? So, you know, the bottom line is that in gene networks, we have problems of huge networks, huge sets of parameters, huge base spaces. But I believe that these can be addressed using methods that relate the underlying logical structure and dynamics. And the real underlying puzzle to me is whether or not these ideas of combinatorial control and simple logical circuits, which really go back to Foucault and Monod, really the grandfathers of this entire field, whether those basic ideas are the way things are really working or whether they're not the way things are working. So as I've gone along, I've tried to mention the names of several of the people who I've collaborated with, and I've had great benefit from having financial support from various agencies. So thanks very much.
Questions?
So correct me if I'm wrong, but there seem to be two notions of robustness. In the yeast cell cycle network, it was robust in the sense that if you flipped the switches, it would attract back to the stable trajectory. Your ant network is robust in the sense that you can change the network structure or parameters and still get an ant. That is, that the trajectory will still be there even if you change some features of the network. Are those in fact different, or are you saying that they're somehow the same thing?
So I think that they're close, but maybe a little different. I think that one of the amazing things in real evolution is that the underlying networks are amazingly robust. All of the different species of ants have the same phenotype. So yeah, so what you're pointing out, which is a good point, is that maybe you can flip some of the genes in that network.
But here, the thing that you're flipping is the transition table and not the states.
Yes, I think that that's right. Yeah. So issue of developmental system drift is something which I think is interesting, which I do not know very much about. So the issue of, you know, what's the concept of robustness and how does it interact with that, I think is something that I haven't thought a lot about, and I think it's very interesting.
Yeah.
So when you're talking about those ends, yeah, it's the same. When you're trying to infer those networks based on these huge sets of genes and many spaces, how do you know that you're not stuck into different local minima that would give you this? So basically, you have like different network for the same phenotype, but in reality, you actually have a similar network for all of them, and you're just inferring some local minima that it's different across the different ant species.
Wait, are you talking about the real organisms or the evolution in silico or
in silico? Yes,
in
because
the analog system?
sorry,
in the analog system?
no, on the ants, right?
What?
On the ant species that we're talking about.
On the ant species.
Yes.
So, wait, you mean when we're trying to find the network that can generate those?
Exactly.
Yeah, I don't know particularly that that's really the right network.
Mm-hmm.
Okay. We, you know, if you talk to the people who studied the biology, Jaeger and Reinert specifically, from this pattern of activation, you can actually do a lot, but then when you want to talk about the activation or inhibition, there are many different networks, much to my disappointment, which can give the same pattern.
Exactly.
But then what they insist on is that, okay, but we have biology which can take us one direction or the other, where people actually study the activation, you know, the actual transcription factors and how they control other production of other transcription factors. So there is not a uniqueness in terms of doing the inverse problem, but the inverse problem can get you to things which are consistent with the data.
Awesome. Thank you.
This might be a naive question. So I was wondering if you are evolving for two distinct functions using to evolving for a network. And would you be able to maybe identify some like distinct motifs where you can add together such that you will evolve for you know, the both phenotypes or let's say functions? I don't know if you
So you're
understand
talking
it.
about evolving a model system
Yes,
to have two characteristics.
right. And
We've never tried it.
okay.
So
Do you think there might be motifs existing for each function that might be addable to a more complicated networks?
yeah, I think that it's an interesting suggestion. You have two things to evolve a network to.
And is it possible computationally to evolve two things at the same time?
Well, I guess so. I mean, what we do is in ours, we just said if the period is equal to or longer, then you keep that particular set of mutations in the ones and zeros. So I guess you would have to give some weight function to two different characteristics, and then just evolve according to that.
And
So, I mean, you're suggesting that there might be a possibility that if you started out with a bigger network, that you might get subnetworks. One subnetwork is associated with one thing and the other with the other thing. I mean, it would be very interesting if you actually just did it and you started seeing that. That's a really interesting idea. I like that idea.
Hi, thank you, Professor. This is a marvelous talk. I just have a question for the students. How many of you have even heard of Jacques Monod or read that paper? Okay, a few. So I think what we're going to do is read a few of those.
Oh.
I'll pull them out. I had to do it.
See, this is the problem of being old, that you remember
And so
all of
those were the
old
greatest.
papers.
They were the most inspiring papers. They were published in the Journal of Molecular Biology in a series starting in about 1961 through 1965. Jacques Monod, who won the Nobel Prize, Pierre Changeau was part of that, and Jeffries Wyman. I mean, there was a lot of nice papers there, classical papers. So we'll pull those up, and I'd like to have a little seminar on those. So you have given us a great trajectory. I mean, Jim Collins has visited with us several times. We learned about the repressilators. Stu Kaufman came here all the time because of the Santa Fe Institute and John Holland and everybody. So
that's right.
you have this marvelous perspective. So where's this field going in the next 10 or 15 years, given your perspective? What's the trajectory of all
I mean, so first of all, the bioinformatics is unbelievable. You know, the amount of information you have in bioinformatics is unbelievable. The task of determining the transcriptional network is fantastic, I think. I mean, the question that fascinates me most is the one that I talked about towards the end, which is how do real gene networks evolve?
Yeah.
And how do you, for sure, I mean, you think about the heart. The heart is unbelievably robust, both in a given individual for some number of years, depending on how long each of you has been there, and also evolutionary. How is that evolving? Is somebody talking about it?
Right,
I don't
and
know.
you see those cardiac myocytes, and they beat, a single cell beats, and
Absolutely.
then the whole sarcomere beats, and then the
Absolutely.
tissue beats and
No,
twists.
but so one of the things about the cardiac, which is something that I actually know more about than gene networks, is the mathematical models in cardiac electrophysiology are based, most of them are based on Hodgkin-Huxley type models, where there are a number of ion channels and there's currents associated with different ionic species through those. When people develop those models, they have to assess the number of different channels, the magnitude in front for all of those channels. And the models are very fragile with respect to those particular currents. But the cells are very robust. So the issue of what's controlling, related to the things that in decrease, and what's controlling the expression, but in particular cell lines, what's controlling the expression of channels in cardiac cells? What's the feedback loop associated with that? I don't think people are studying that. So the thing that I found, well, next week I'm going to visit the colleague, Enrique's colleague, and we were talking about a problem that I was very interested in that I know nothing about, which is the evolution of the karyotype.
Yeah.
How does the karyotype evolve? It's related to the evolution of the genetic circuits also. So to me, that's also interesting. So, there's also the comparative genomics, the comparative control. So I think that all of these, there are lots. And then the other thing that I was mentioning is this really the way things are working? What's the role of histone modification? What's the role of RNA in the control?
Right.
So is RNA acting to help being an on-off switch? If you really have the control of on-off, is it just by rather weak sigmoids, or they have the kind of very strong sigmoid functions like your colleague Chong found with Ferro, where you have these incredibly steep sigmoids? What's happening in the gene control? I mean, it seems like there's almost a universe opened up of things that can be done which were not possible 40 years ago or 30 years ago.
But other thing is that maybe the data the real data point of view, we cannot experiments only we construct like 2-3 time points. So that's the way we can use mathematics to reconstruct the things we're not observing to complete the trajectory.
Okay, well, you will figure out ways to improve the time resolution.
Yeah.
So, you know, time resolution, there's some very strong
Exactly.
time resolution in control of endocrine oscillation. Yeah. So, you know, I think that there's need for better data, and you're working to do it.
Well, thank you so
Okay,
much.
great.